So, I'd like you to start off by checking your vocabulary. This unit does have a lot of vocabulary, and you're going to have to be practicing it in order to comprehend the processes and know all of the understandings that you're supposed to know. So we've got an image here, and what is this showing? Is it showing two separate chromosomes? Is it showing what we call a bivalent? Is it showing one pair of sister chromatids? Or is it showing homologous chromosomes? And the correct answer would be two separate chromosomes. Okay, it's clearly not a bivalent because the bivalent would be the homologous chromosomes that are lined up with each other next to each other. And since these are also not homologous chromosomes, and how do we know they're not homologous? Well, they look quite different. They have different lengths. They have different alleles as indicated by these bands over here. And so they're quite different from each other, so they're probably not homologous chromosomes. They are, however, two separate chromosomes. Another question, feel free to pause and try to answer on your own. So this image is showing two bivalents. The reason why this is showing two bivalents is because we have the two pairs of homologous chromosomes lined up with each other. Okay, they are not 8 separate chromosomes because we can count when they're connected here at the centromere. The sister chromatids that are connected at the centromere are considered one full chromosome. So we've got 1, 2, 3, and then 4 chromosomes, not 8 separate chromosomes. 2 pairs of sister chromatids. Well, I would say that this is one pair of sister chromatids. This is another pair of sister chromatids, so that's 2 pairs right there. But then we've got another pair here of sister chromatids, another pair here of sister chromatids, so that's actually 4 pairs of sister chromatids. Okay, why are they not 2 homologous chromosomes? Because we actually have 4 chromosomes. So 2 homologous chromosomes would be these 2 chromosomes, right? These are also homologous chromosomes. There's 2 of them. 1, 2. I'm going to go back to this first slide so we can see what have we actually covered so far. Chromosomes replicate in interphase before meiosis. Yes, we've talked about this. We've covered it. How about crossing over is the exchange of DNA material between non-sister homologous chromosomes? Yes, we've covered this. Crossing over produces new combination of alleles on the chromosomes of the haploid cells. Yes, we've covered that. Chiasmata. Formation between non-sister chromatids can result in exchange of alleles. Yes, we've talked about that structure that forms during crossing over between the crossing sister chromatids. Next, we've got homologous chromosomes separate in meiosis 1. That is correct, and we have covered that as well. Sister chromatids separate in meiosis 2. Correct. That is what separates in meiosis 2. Independent assortment of genes is due to the random orientation of pairs of homologous chromosomes in meiosis 1. Okay, so we haven't completely talked about what independent assortment is. We did talk about random orientation of the chromosomes, which results in independent assortment. That is true. Draw diagrams to show chiasmata formed by crossing over. We can do that as well. And let's take a look at the guidance statement because sometimes that can be useful and give you some clues as to how you should be studying this. Diagrams of chiasmata should show sister chromatids still closely aligned except at the point where crossing over occurred and a chiasmata was formed. Okay, so let's finish off these last few slides. This one is a review slide where it's showing the sources of genetic variation and what promotes genetic variation in meiosis. Those two things are crossing over and random orientation of the chromosomes. What do we mean by crossing over? crossing over each other and exchanging genetic information. This helps to produce different varieties of gametes, and so does the separation of the randomly oriented chromosomes. So I'm going to go into more detail of random orientation in a second. Okay, so if we're looking at this slide here and you click on it, it opens up this video. It has no audio to it, but it's kind of useful in just skimming through to visualize meiosis and also the random orientation of the chromosomes. So here we've got different alleles marked on different chromosomes. We have a big S marked on this chromosome, we have a small s marked on this chromosome, a big Y allele marked on this chromosome, small y allele marked on this chromosome. So let's play the video.
With just 2 pairs of chromosomes to illustrate a genetics concept called independent assortment. We will track the movement during meiosis of the cell's 2 pairs of homologous chromosomes. Before meiosis begins, the cell synthesizes new DNA and thereby replicates each chromosome.
Okay, I lied. It does have audio, but anyway, that's actually useful. It is replicating, right? And now we can see the replicated sister chromatids on each of these chromosomes. And so there are 4 different chromosomes there.
Early in meiosis, the pairs associate. Note that the chromosomes in a pair are not quite identical. In one pair, for example, one chromosome carries a recessive allele of a gene, such as a little s, and the other carries a dominant allele, such as a big S. The 2 pairs line up at the cell's midplane during metaphase 1 and may adopt one of 2 alternative alignments. The alignments provide the basis for independent assortment.
Okay, so this is the random alignment of the homologous chromosomes. Notice that they are different. Over here on the top, we have an alignment between small s on top, big S on the bottom for the alleles. We have a big Y on the right side on this pair of homologous chromosomes, big Y allele on top, small y allele on the bottom. Over here, it's different. We have small s, small y, big S, big Y.
Cell may adopt alignment 1, in which the little s and the big Y alleles end up in the same daughter cell. Or the cell may adopt alignment 2, in which the little s and big Y alleles end up in separate daughter cells. From a large pool of cells that initiate meiosis, on average, half of them adopt alignment 1, and the other half adopt alignment 2. During the second half of meiosis, the 2 cells from each alignment divide into 4 haploid cells. The result is an equal number of the possible genotypes: little s, big Y, big S, little y, little s, little y, and big S, big Y.
Okay, so alignment 1, this is showing what one random alignment of the homologous chromosomes could have produced here for their 4 gametes that were produced. So you can imagine that these are, for example, sperm cells or egg cells, although there is a discrepancy about egg cells with meiosis and with higher level that we will get into later. Okay, and this is another alignment that could have happened that produces different gametes. So it's increasing genetic variation in the gametes that are produced. This is going to cause there to be greater genetic variation in their offspring, which is a good thing. This is what we want for the survival of a species, for them to have more genetic information.
Because there are equal numbers of genotypes, we can say that the alleles of the S and Y genes assort independently during meiosis. Note that alleles of different genes always sort independently if the genes reside on different chromosomes, but not necessarily if they reside on the same chromosome.
so what is the difference then between random orientation and independent assortment? Random orientation depends on these alignments that we were talking about in that previous video. The two different random alignments which caused variation in the gametes that were produced. Independent assortment is the actual behavior of those alleles assorting and separating into the different gametes independent of one another. So, for example, alignment 1 and alignment 2 produces gametes that are different from one another. This means that the small s and the big Y are not necessarily linked to each other in any way and can independently assort into different gametes. The big Y and the small y, or the big S and the small y, also are not necessarily linked to one another. They can independently assort into different gametes randomly. Most genes are not linked to one another, and they will independently assort and go through crossing over. However, there are some anomalies that rarely do show up, where genes always show up together. For example, you could have fruit flies where the red-eyed trait in a fruit fly is linked to these larger wings. And so whenever you have red eyes, you're also going to have the larger wings. This is another example of another fruit fly. Possibly the white eye trait is linked with the folded wing trait pattern. So linked genes do not independently assort from one another. They remain connected to one another and always show up together. Again, this is an anomaly. This rarely happens where genes are linked to each other. The reason why I'm mentioning this is because it is in Nature of Science, which is connected to TOK and anomalies that happen, maybe paradigm shifts or learning new information, or how do we know the new information. But I'm also explaining this because you will see this actually later. We will talk about linked versus unlinked genes and what the genetic patterns and the frequencies of those end up being and how to recognize it. Okay, so now we've covered this one. Independent assortment of genes is due to the random orientation of pairs of homologous chromosomes. We haven't done drawing diagrams to show the chiasmata formed by crossing over, so let's do that. Okay, so if we have one chromosome and we have the homologous chromosome of that one chromosome, it would be right here. After it replicates, remember, it's going to be looking more like this, right? Okay, so now we have the replicated homologous chromosomes. We have their centromere region right here. Okay, what we're supposed to be showing is the chiasmata structures that can form. So the way I would suggest doing this is just like so. That, potentially. And you could draw the exchange of information within the chromosome. Maybe if this had already exchanged information, it would be a different color. Right? We have the random exchange of information that occurred between the homologous chromosomes and the non-sister chromatids. All right, that's it for 10.1.